First, I wish to thank the organizers for giving me the opportunity to present my work. In 2 sentences, I will present myself. My name is Shula Shazman. I did my first degree in computer science at the Technion, and then did my PhD in computational structural biology at the Faculty of Biology at the Technion. Traveled to a postdoc at Columbia University. My topic of research were DNA binding proteins, the RNA binding proteins in terms of structural features. Today, I'm a member of the Faculty of Computer Science in the Open University, and my topic of research are health informatics. So, autism spectrum disorder, ASD, is a complex neurodevelopmental disorder with strong genetic basis. Yet, only a small fraction of potentially causal genes are known with strong evidence to the relation of autism. The method development in the study, the method I developed and I will present in this study today is a deep learning approach to classify ASD causal genes based on the intrinsically disordered regions of their product proteins. Autism spectrum disorder is called a spectrum since the strength and challenges can vary widely from person to person. Biologicals are, researchers are on their way to find genes that relate to autism. We are talking about genes because they allow us to find the original problems of the problems. The current genetic knowledge regarding ASD causal genes is found in several databases. 2 of the major databases are shown in this slide. Autism Knowledge Base and Simmons Foundation Autism Research Initiative. In both databases, risk genes are scored based on the evidence supporting their linkage to autism. Autism KB contains 198 human genes with high evidence to the relation of autism, while SFARI database contains 207 human genes. However, the data in both databases is representing only a small fraction of the estimated 400 to 1,000 likely genes to be involved in autism suspectability. To reveal a more comprehensive genetic knowledge of autism, we need complementary approaches to reveal additional potentially genes and to do a genome-wide prediction for ASD causal genes. A previous study presented a complementary machine learning approach based on a human brain specific gene network. This method predicts hundreds of candidates for which there was minimal or no prior evidence to the link or relation of autism. Nevertheless, the genetic heterogeneity underlying ASD is still frightening, and as a result, it is unlikely that all candidate genes will be revealed using sequence studies, network, or statistical association alone. So, the main source knowledge regarding potentially ASD causal genes is found in sequence studies. I was thinking it would be interesting to find out whether there are structural features in proteins that are the product of ASD genes. And one of the protein structural features I was curious to search its relation with ASD was intrinsically disordered regions. Intrinsically disordered regions are part of proteins that lack a stable 3D structure. It is well known that intrinsically disordered regions are overexpressed in disease pathways. For example, nearly 25% of the disease-associated missense mutations are located inside intrinsically disordered regions. Furthermore, intrinsically disordered regions are expressed very much in proteins that are involved in cell signaling, such as ASD causal proteins. Another study investigated the relation between protein involved in ASD and high content of intrinsic disorder. Approximately 80% of the proteins exhibited long regions of disorder, and about 30% of the single nucleotide variation were located in disordered regions. The x-axis of the histogram shown here is the number of amino acids, while the y-axis is the fraction of proteins involved in several neurodevelopmental diseases like schizophrenia, ADHD, bipolar disorder, and autism spectrum disorder. The brain bin is a group of proteins which are expressed in the brain, but not involved in none of the above mentioned disorder. Interestingly, you can see that the column of the ASD, which represent the percentage of proteins expressed long regions of intrinsically disordered, is the highest compared with the control group of the brain and compared with other neurodevelopmental disorders. Therefore, because of the need for complementary approaches for genome-wide prediction of autism risk genes, and based on the studies which suggest that proteins involved in ASD present high content of intrinsically disordered regions, I developed a classification method to predict ASD causal genes based on intrinsically disordered region. The method used artificial neural network, and I thank to Marina in the previous lecture, which gave insight into this method. And the method successfully distinguished between proteins involved in ASD from proteins that are not involved. The process of building an artificial neural network classifier is composed of 4 major steps. Step 1, gathering the data from several resources. Step 2, cleaning and preparing the data to have homogeneity. Step 3 is training and model the data. And step 4 is training and testing the data in order to get insight and to learn some new results. The first step was getting the data. I selected 198 genes found in SFARI database with high score of category 1, and also found in Autism KB with score 16 or higher. The group, the control group, or the group for genes that are not involved in ASD contained 245 proteins, which were not found in SFARI and have a score of 0 in Autism KB. The final data set contains 2 lists. One is the ASD causal list, which, as I mentioned, had 198 proteins, and the control list with 245 proteins. Each protein has a vector of 17 features and 1 class feature, which means if the value is 1, this is a ASD causal gene. And if the value is 0, this means it is not an ASD causal gene. Among the 17 attributes are the number of intrinsically disordered regions, their length, helix, beta sheet, aggregation, serine threonine, and more secondary or structural features, all expressed in percentage. As shown from this slide, there are detailed distribution for each one of the features for the two classes. Color blue is the non-ASD causal genes, while color red, what of? Sorry. Yeah, sorry. Color blue is the ASD causal genes, and the red is no ASD causal genes. What can be seen from this histogram that there is no significant difference between the distributions. So we can conclude that none of the features can predict alone the connection or the relation to ASD. The third step is model the data. As I mentioned before, I use an artificial neural network. The best architecture of the network is shown here in this slide. It contains 4 hidden levels with 9, 9, 19, and 19 neurons respectively in the four hidden levels. The activation function used for this model is the sigmoid, which is a good activation function to differentiate classes with values of 0 and 1. In addition to artificial neural network, I use several decision tree classifier, the J48, the logistic model tree, random forest, logistic, and in addition, logistic and naive-based multinomial classifiers were tried. We arrived to the fourth step. In order to test the data, 2 test approaches were used, the tenfold and the leave one out test. As can be seen here from the results, the accuracy of the artificial neural network was the best, with 73% for the leave one out. When we are talking about different classifiers, accuracy is not enough to compare the result. We need another estimator, at least one, which I present here. Here is the area under curve. And again, the best results here were with 0.82 for the artificial neural network. It is interesting to note that by visualizing the tree generated by J48 model, as shown in this slide, it can be seen that the most effective feature to differentiate between ASD and non-ASD causal proteins is the number of regions with more than 30 amino acids. The logistic model tree was less successful than the J48 and the artificial neural network, but still we can conclude something interesting from the correlation of the features, of the weight of the features, because in logistic model tree we get weights for the features, the correlation between those weights and the different classes. And again, we can see that the most significant correlation is with the feature that contains the number of intrinsically disordered region which contain more than 50 amino acids. The correlation is positive with the ASD causal protein, colored green, and is negative with the ASD non-causal genes, colored red. To summarize, this study presented a computational approach of genome-wide prediction. The method uses an artificial neural network and is based on structural information of the product of gene, the proteins. The method successfully distinguished causal from non-causal ASD genes with area under curve of 0.82. The most important attribute for classifying ASD causal genes are related to the number of intrinsically disordered regions and their length. The result indicates that the contribution of intrinsically disordered region can be taken into consideration when trying to disappear the mechanism of autism. I'm currently working on update data of Safari because they do have a lot of updates, and to perform a genome prediction, because all I did is taking the sets I told you and trained and test, and I would like to do a genome wide prediction for all genes expressed in the brain, at least. Okay, so some more reference which have other complementary approaches for identifying intrinsically causal genes or proteins. And I wish to thank the Open University that support my work, and I wish to thank you for listening.
Hi.
Yeah, sorry. Yeah.
Thanks for a nice talk. Sorry to ask such a basic question, but could you define what you mean by intrinsically disordered protein in this context? Like how is that defined?
Intrinsically disordered regions?
Yeah.
Yes, I just passed through it, but intrinsically disordered regions can be defined by several methods. Regions inside proteins. Just a minute. I choose you see the first reference here. This is, again, a machine. Actually, the first basic way to define intrinsically disordered region, when you look at X-ray of structure of protein, the missing parts are intrinsically disordered regions. So the first reference took huge data from the PDB and used machine learning approaches to predict where and why. Because we know the sequence of the protein, why don't we see in the X-ray the structure? So I choose that reference for yeah, sorry. Yeah.
Thank you. I guess that the 200 candidate genes that you tested for were genes with various variants causing autism. Did you look at GWAS results before moving to whole genome? Because there you will have many more.
Yes, they already asked me that question. I think I will. I haven't, but I think I will. Yeah, thank you. Yeah, it's a good question. Suggested.
Any more questions? Thanks, Shula.
Okay.